Hello and welcome to today's webinar. My name is Melissa and I will be your host for today. This webinar is part of our regular Wednesday webinar series, and you can find out more about that on our website. In today's webinar, we're going to hear from Nikki Karamenis, who's going to tell us more about Data Visualization 101.
So this is an introduction to data visualization with a particular focus on design scientific figures. I'm Nikki and I'm a senior user experience designer here at the EBI. Specifically, we're going to go through who is the seminar for and what is the recommended reading for you. And I'll talk a bit about why do we need to visualize data. And then I'll focus on two important aspects of human perception and how we make quantitative judgments and how we perceive color. And I close with some thoughts about how we can deal with complexity in visualizing data. You will notice that in most of the slides, I spell visualization with S, which is the British spelling. But we also have text from others where they spell it with Z, which is the American spelling. This particularly in titles of books and figures and so on. And I'd also like to mention that the field is abbreviated as data viz, and this is sometimes spelled with S and sometimes spelled with Z, particularly in social media. So when we put the seminar together, what we had in mind is a person like Pat, who is an expert in a particular type of biology, but doesn't know that much about data visualization. So her question is, how can I best communicate my research results? And her audience is her supervisor, people that she works with in the lab, and other wet lab scientists in her area. She mostly writes lab and conference presentations and posters, and some journal publications. And she reads high-impact journals like Science, Cell, Nature, with the aspiration of publishing her own research. And it was in one of these journals, particularly Nature Methods, where a group of leading data visualization researchers got together between 2010 and 2015, and they published 38 one-page articles on particular aspects of data visualization. So the webinar today is really based on material from them, and also a few other selections that I have made. You can find all these articles in Nature Methods itself by following this link, and they also sell it as an ebook through their shop. So why do we need to visualize data? Napoleon said that a good sketch is better than a long speech, and in English we also have the saying that a picture is worth 1,000 words. So we'll go through an example of that. Imagine that you have this data. So these are 4 data sets that have identical means, standard deviations, correlation, and regression coefficients. So in terms of the summary statistics, they're really the same. But your job is to communicate whether there are any differences between these 4 sets of data and whether there are any outliers. So you would just present a table and start talking through the table and ask your audience to follow you when you point out particular aspects of the data set. But this really requires a lot of work from their part. You have to give a long speech. On the other hand, if you just plot the data, the differences and the outliers are self-evident, almost self-evident. So in that case, you rely on your audience perceptual abilities. They can very, very quickly look at the 4 plots and see the differences that they have, and also very easily identify the outliers in each of them. So this difference between perception and cognition is a very important one, and this is a fundamental reason why we want to visualize data very often, because it's really much faster and requires much less effort for people to use their perceptual abilities in understanding data as opposed to their cognitive abilities. Generally speaking, we visualize data to communicate our findings, but there's also an area of visualization that has to do with exploring and formulating hypotheses. Today, we focus on visualization for communication, and as I said, mostly we'll be looking at static graphs, the kind of things that you put in slides, publications, and presentations. So as I said before, there are 2 main areas of perception that are very important for data visualization. One of them is how we make quantitative judgments, and the other one how we perceive color. So we look at judgments first. Cleveland and McGill ran a study in the mid-80s where they effectively asked people to tell them how many times bigger A is compared to B. And they used different ways in which they encoded this difference. So you you, for example, place items in a common scale and ask people to tell you how far away these 2 points are on the common scale. Or you try to shift the scale. So again, the items are further away from each other, but they don't have a common reference, the 2 scales. You can encode the values as lengths of lines and ask people to tell you whether this line, how much bigger this line is compared to this one. Or encode it as an angle. So try to tell them you're asking people to tell you in that case, how much bigger this angle is compared to the one above. Or encode it as a circle. So in that case, the question is how much bigger this circle is compared to this one. Or as a sphere. So what people are trying to judge now is the volume of the sphere. Or as different shades of the color. So in that case, you're trying to ask people to tell you how much darker this shade of pink is compared to this one. You could also have color schemes that consist of different hues of different types of colors. So what people need to do then is to try to depict how much more this darker green is compared to the blue with reference to this particular scale. So what Clifton and McGill did is they gave people these different encodings of this question, and they measured how accurate people were in their answers. And they found out that when you use a position in the common scale, people are much more accurate in their judgments. And as you move along, so if you shift the scale or use length, angle, and area, people become less accurate. So this is known as the ordering of the perceptual tasks. And this is the main reason why data visualization people in general are not very fond of pie charts. So if I give you this pie chart and I ask you whether A, the area represented by A, is more than the one by D, you really have to judge the difference between the angle of this area here, or the slope of the line, or the actual area itself. And for most people, this is actually quite difficult to do accurately. On the other hand, if you plot the same data as a bar chart, you can see much more easily that D is actually a bit more than A. And it's much easier to estimate how much more this is. So to me, this looks something like between 5 and 10%. There's a person called Edward Tufte, who is one of the pioneers in data visualization. And he wrote a book called The Visual Display of Quantitative Information. So if you go to page 178 in this book, you'll find this quote: "The only worse design than a pie chart is several of them, and the pie chart should never be used." This is part of a wider quote, but you can see that sometimes pie charts can be very heavily criticized in the data visualization field. So if we're not supposed to use them, what can we use instead? It really depends on the goal that your visualization has. So we're going to go through 3 examples where we actually show the same data, but we try to convey different information. So imagine that you have 5 categories. This could be species, and 5 items. This could be conditions for the same experiment. If your goal really is to find out which item scores the highest in your scale, then you can just plot them as bar charts. And if you want to depict, so in that case, you see that item 1 has a higher score, right? And progressively, it gets reduced. And you can also depict how much each category contributes to the item, but then it's quite difficult to make relative judgments. So for example, if you focus on the green area here, it's quite difficult to tell whether the values of the green category actually change across items. So now if your goal is to depict that, you try to understand the differences in each category across the items, it's better to align them in a common scale. So the example that I gave before, the one of the green charts here, these are items in a non-aligned scale. If you put them on the same reference, on the same scale, you can see very easily that it starts, the score starts being just above 15, and progressively goes down. It's much easier to make these quantitative judgments in this type of visualization. On the other hand, if you want to compare which category contributes most to each item, so for example, if you want to figure out whether the pink bar here is higher or lower than the red one, then you can use the group bar charts. And you can see that indeed it's a bit bigger, and it's much easier to tell the difference between the 2 data points. So what you actually plot depends a lot on the message that you want to convey, and this is something we go back to later on as well when we talk about complexity. I'll now talk a bit about color, and particularly how we perceive color. And we need to start by actually defining what we mean by color. Color has three properties. The first one is hue, so that's the difference between, for example, red and blue, and this is what we refer to when we say color, the word color in everyday language. But it has two more properties that are very important from the visualization perspective. One of them is saturation, which is how much of the actual color you have. So if you look at the end of the second row, at the rightmost square, this has more velvet color than the rest, so it's more saturated in this particular color. As you move towards the left, it becomes less saturated. And there's also an aspect called luminance, which has to do with the brightness of the color. So typically black is the darkest color, and you can make it brighter by adding white. So the first thing to remember now is that, as we said before, if we go back to this ordering of perceptual tasks, color hue, so the difference between colors, as we say in everyday language, is not a good way to encode quantitative data. It's very, very difficult for people to make judgments about whether blue is more or less than green. And this is the reason why, again, data visualization people are not very fond of the so-called rainbow color scheme. This color scheme has two problems. One of them is that it's perceptually not linear. So if you look at this example here, between the data points towards the end of the scale, you have an increase by 1 unit. However, the color itself changes very little, so it becomes a different shade of green. If you go further up in the scale, you have again a difference of 1 unit, but the hue changes a lot, so it becomes orange from yellow. And this really biases the visualization towards these changes, towards higher up of the scale, although in reality, the absolute difference is the same. The other problem is that these color schemes are not perceptually ordered. So if you ask people to tell you the order of the colors in the rainbow, most people don't really know that. And secondly, as I said before, if you ask them to tell you whether green is more or less than blue, they cannot answer this very consistently. So what is recommended in this case is to use a grayscale. In this particular example, they start with 90% luminance of black to 10% luminance of black. And this is much easier for people to actually depict, and it's easier for them to make this kind of judgments. However, in general, it's better to avoid using color to encode quantitative data. And we're going to go through an example, again from nature methods, of how you can avoid color. So this is a network, and they colored every node in the network based on the correlation score, which is depicted on this rainbow scale. So what they have done in nature methods is they redesigned that using bar length deposition. So the correlation score now is shown as the length of the bar, and you can see that it's much easier to identify the items that have the highest correlation values and also see the difference between these items. The other thing that happened in this particular visualizations is that they had extra information. There were other elements. I think in this case these are enzymes that have something to do with this biological process that is depicted here. So they placed those in the visualization as well. So in this way, you can actually depict more data and much more accurately. Another thing to keep in mind is that perception of color varies according to the context. So this image comes from this website at MIT. It's attributed to Edward Adelson. If you look at the squares labeled A and B, most people perceive them as being different color. However, they're actually the same, and this becomes more obvious if you actually join them with the line of the color. And particularly if you focus on the part of the line that it is within the tile, then you can see that the color is actually the same, but it looks different because of the context that it is in. So this kind of visual illusion is actually quite important because it happens a lot at heat maps. Again, this is an example from nature methods, and you can see how the two squares that are labeled with an asterisk seem to look different, but they're actually the same color. So again, this biases your visualization. Another important problem with color used to encode quantitative values is that sometimes important elements don't always stand out. So in the scheme here, they use dark red for the highest values, and presumably what you want then in a visualization like that is for people to be able to spot them, spot this high value items. However, this is quite difficult. So you can see here in the middle, actually, this looks quite hidden. On the other hand, the visualization encodes the low value item with a color very close to black, very dark blue, and our eye is drawn to that. So this visualization is actually biased towards the lowest values instead of the highest values, although the highest values are actually equally important. Another thing to remember is that not everyone sees the same colors, and particularly there is a condition called color blindness, where people cannot distinguish easily shades of red and green. And so I prepared a small quiz, which I'm going to read, and I'll invite you to think about the answer, and then I'll say the answer myself. If a submitted manuscript happens to go to 3 male reviewers of Northern European descent, the chance that at least one of them will be colorblind is either 2, 12, or 22%. So give yourself the time to think. And now I'll tell you the answer, which is 22%. Colorblindness is much more common in men than in women. It's a sex-related, gender-related trait. But this shows that it's actually quite likely to happen. So if your visualization uses green and blue, then it could be a problem for them. This is the standard color blindness test here at the top with A. And you can see what happened here is the hues don't vary in lightness and saturation. So if you turn this into gray, the pattern of 6 is not visible. So this is very similar to how colorblind people see it, except that what they see is actually shades of brown. On the other hand, if you vary the lightness and saturation of the two hues, so if it's not just the hue that is different, but also the lightness, and then you remove the color, the pattern of 6 is actually much more visible. And this is a technique that is used in color schemes that are optimized for colorblind individuals. This is the one that's been suggested by Nature Methods, and you can see the actual color on the first column and how colorblind people will perceive it in the next column. This is protanopia, which is a type of color blindness, and the heteronopia, I think this is called, which is the most common one. So there are other schemes like that. Color Brewer offers one, and also there is the NASA Color tool. And the other thing that we can do if you produce an image is actually to check it using the Photoshop color blindness tools, and also tools online such as VisCheck and WebAIM. And it's quite important because sometimes it's not just red and green that can cause trouble. It can be any color that includes these components. The other thing I wanted you to notice is that this color scheme, which could be used for categories, has 8 colors, and this includes black, which is the color that you're going to use for your type and probably for the lines of your visualization. And you should use at most 6 to 8 colors for categories, and ideally even fewer. If you have more than 8 categories and try to depict them, distinguish them with color, it's not going to work very well. So here is a quick summary of what we said so far. If you have quantitative data, position is the most effective way to encode these values, and color hue is the least effective way to encode these values. A gradient of luminance might be an acceptable last resort. If you have categorical data, you may encode at most 8 categories with an accessible color scheme, which varies luminance, saturation, and hue. So now I'll talk a bit about how we deal with complexity. And this is a question that I get a lot here at the EBI, where we have a lot and complicated data. And typically what I try to ask people is to tell me what is the question that they're trying to answer with their data. So we're going to go through an example. Let's imagine that we're asked to report on the difference between exports and imports in a country over time. And the data that you're given is the volume of exports and imports. So if you plot the actual data, the original data, and you want people to be able to compare these 2 years that I highlighted in red here, it's actually quite a difficult thing to do, because what they have to do is they have to compute the difference between the 2 lines in these 2 data points. And this is again an example where people really need to require to use a lot of cognition instead of perception. It's quite a difficult task. If you think about the question though, the difference between exports and imports, you can transform the data and answer it. So what you really need to do is to compute a differential, which is known as a trade balance in commerce. And you can see that you get a line that looks a bit like this. And in that case, it's actually much easier to tell that in the later year, the trade balance is slightly higher. You also get a different impression of the actual trend. So you can see that the trade balance kind of going up and down. It's actually slightly declining in later years. Whereas if you actually use the original visualization, because this is based on the volume of imports and exports, has an upward trend. And it might actually mislead you thinking that the trade balance itself is going up, where actually it's not. Another technique is to avoid displaying all data in one plot. So this is a graph where it shows the relative abundance of 24 cell lines over time. And this is known as a stat line. So they basically superimpose the lines on top of each other. A better technique is to plot each cell line separately, each item separately. And this is called small multiples. It is another technique popularized by Edward Tufte in his book. Because these are in a common scale, it makes it easier to compare items with each other, and you can see just from the shape of the plots what the patterns are. There's a variant of small multiples which is called spark lines. So these are very small graphs that depict trends, and they often appear in tables or even in text. Here's an example of the influenza of the flu cases in 2012 and 2013 in a number of countries. You can see again, as this is embedded in the actual table, it's easier to depict threats. So for example, if you take Canada, you can see that there was a peak earlier in the year compared to Australia in 2012, whereas in 2013, actually the actual pattern of cases was very different, even though the absolute number of cases was higher. So these are the kind of judgments that you can make based on data visualized like that. We talked a bit about heat maps and the differences, the problems that we have that they introduce because of the use of color. And there are two techniques that we can use to overcome those. One of them is clustering to reveal patterns in the data. So what they have done here is they clustered values that are similar to each other, high and low. Another more powerful technique is to add gaps to make the clusters and the outside outliers more obvious. This type of visualization is called the gap map. And there is a tool at the Broad Institute called the Morpheus, which produces maps like that. What is interesting in this gap map from a visualization perspective is that it uses whitespace and grouping to structure the data. This is based on a set of principles called Gestalt, and one of them says that items that are closer to each other tend to be perceived as belonging to a group. This is why the dots on the far left are seen as belonging to lines, because the distance between the dots horizontally is smaller than vertically. On the other hand, these dots here are perceived as belonging to columns because the distance vertically is smaller than the distance horizontally. Another very good use that the gap map does is that it uses white space to emphasize an allow breathing space. So effectively, this is what draws our eye on the red, the blue isolated points on the top row, the so-called, which seem to be outliers. And this is similar to how we perceive the pattern. This is one of the 4 data sets that I showed you earlier in the presentation. So you can see that we're drawn towards this dot because it's surrounded by white space. One of the things that they mention in this article about negative space in H2 methods is to really treat white space as a commodity and try to use it for emphasis instead of actually filling it in with more content. It is really an opportunity to keep breathing space to your visualization and emphasize the things that are more important. There is an alternative to the heat maps, which is called the parallel coordinate plot. And so here's an example of that. This is a typical heat map. The dark areas are very high values, and the brighter ones are the gray ones are lower values. We have 2 types of categories, and each row is values over time. So what the parallel coordinate plot does is the value is depicted on the y-axis, and the x-axis is still the time. So effectively, each row in the heat map becomes a line on the parallel coordinate plot. And this has the advantage of, first of all, it uses position, so it's much easier to actually value what the absolute values are. It's much easier to judge what the absolute values are. It's also easier to make comparisons, for example, between the first line and the lines underneath it. Although it has a disadvantage that sometimes it can become very, very dense. So you can see that towards the middle of the visualization, there is actually a cluster of lines. It's quite difficult to follow the lines. There are techniques that you can use if you want to emphasize a particular line, and we'll show one later on. Another thing that's interesting in this type of plot is that they use the principle of connection. So I talked about the Gestalt principles before, and specifically proximity. So things that are closer to each other are perceived as being grouped together. And this is stronger than the principle of similarity. So if you focus on the squares and the circles on this slide, you will see we tend to think that the square and the circle are more of a group than the two squares together. And this is because the square and the circle are closer to each other in the row rather than the column. Connection is even stronger. So if you draw a line between these items, you override the principle of proximity. And the closure is even stronger than that. So again, you see these two things as being connected because they are framed, even though there is a line between them. So the heat map, the parallel coordinate plot, effectively replaces the heat map by using connection to connect the data points. This can also be misused. So this is an example from Tamara Manzer's book called Visualization Analysis Design, where they basically plotted the average height of men and women, and then they connected the two dots with a line. So when they gave this to people and they asked them to explain what the visualization shows, people said that the more man somebody is, the more male somebody is, the taller they tend to be. So these two distinct categories were perceived as being related because they were related with a line, they were connected with a line in the visualization. So this again can be misleading. A better way to depict this kind of information is just by using bar charts and showing very clearly that these two categories are distinct. Another nice thing about this particular parallel coordinate plot is that they use the guide, so the vertical lines here on the x-axis, to guide the eye. By contrast, sometimes this kind of grids become very thick, so there is a lot of ink that is used, and this actually hides the actual data. So this is an example again from one of the nature methods articles. Edward Tufte came up with the concept of data ink. So what he said is that basically most of the ink should be used in visualization to show data, and here just to maximize actually the ratio between data and ink. And one way to do this is again by making the grids thinner, visible enough to guide where the eye should go, but not including data. Tufte also talks about a concept called chart junk, and these are graphs like this one where we have a lot of redundant color, shapes that are not actually particularly meaningful, and also textures. Particularly, this tend to appear a lot on bar charts. And these not only are not meaningful, so they don't really introduce any information, they can actually be confusing, they can be distracting, and again, they can bias people towards particular interpretations of your visualization. So he really suggests that we avoid doing this unless they have a particular purpose. Another source of complexity is how to depict sets. A very common visualization is Euler diagrams. So the intersection between sets, in this case, is shown by the intersection of circles, and the area that its intersection covers is proportional to the number of items in the set. So this works all right for up to 3 sets, but if you have more sets or if you have more items, it kind of breaks down. So for example, in the example here, there is an area that has 4,000 items, and this is very small. And the one next to it has 450 and it's much larger. So the analogy really breaks down. There is an alternative visualization called the matrix layout that we can use for this type of sets. So in this example here, we're looking at the overlap, these are the mutations of particular genes, and the intersection is shown by the connection between the dots. So the one that I'm pointing at here, the fourth one, is the intersection between EGFR, P10, and the number of items is depicted by the height of the bar chart. So that there were 16 patients in this case that had mutations for those two genes. You see that this visualization, the bar charts are ordered according to the size, so it's very easy to see which is the highest set. And the other nice thing that it does is that it shows the empty sets as well. So there are a number of possible intersections that actually don't have any items in them, which is something you cannot really depict directly in an Euler diagram. This is also called the upset plot, and you can find it online in this URL. It was introduced by Alexander Lexer and his colleagues. Another particular troublesome aspect of visualization is node link diagrams. So this often become hairballs, and you see a lot of lines overlapping and also items hiding each other. One way to deal with occlusion is by forming an adjacency matrix. So what happens in this case is that each node becomes a row and a column, and then the connection between the nodes are shown by the shaded areas. So this gives rise to patterns like this one. So these are elements that are very highly interconnected with each other, the so-called clusters, so the blue elements in this visualization. And you can also depict hubs, which are nodes which are connected to other individual nodes, which are shown as rows or columns in this visualization. It is more difficult you cannot actually follow the path of a matrix in a visualization like that, but it makes it easier to see where are the areas of high concentration. Now, this kind of data are multivariate. They have a lot of dimension, and particularly if we have 3 dimensions, it seems to be quite tempting to use a 3D chart. This is again not a very good idea because it introduces the same problem. So it's quite difficult to judge what is the actual values in these charts, because effectively they are 2D. And also there are issues of inclusion. 3D visualizations are helpful for things that are inherently 3-dimensional, like structures, for example, but not for abstract data. So the right thing to do in this case is to make 2D charts and sometimes combine them. A very popular visualization of several dimensions is a scatter plot. So here we have 4 aspects of the length of the growth of the plants and 3 different types of plants. So you can see that you can do pairwise comparisons between them in this visualization. And you can identify the particular clusters, so the different types of plants kind of show quite clearly as being distinct. And also, if you plot it as a parallel coordinate plot, then you can see the data points of each individual plant. So the red line here is effectively one plant, and this corresponds to the dots that have been highlighted here. So this is an example of a linked visualization where you highlight a particular point, and then you show it from different types of views. And there is a tool called Celebra that does the same for networks. So there is this tool has a large network view, and then small multiples of parts of the network, as well as a parallel coordinate plot for expression values, I think, for pathways. So the idea is that you really look at the same aspect, the same types of data, but from different perspectives, in order to identify potential relationships between the data sets. So here are a few ideas about how to deal with complexity. First, plot what matters most to answer the actual question. This is the thing that I find most people really forget when they think about the visualization they want to create. Don't display everything in one plot. Small multiples of spark lines can be alternatives to that. Use whitespace and grouping to structure and emphasize. We looked at the gap map as an alternative to an unstructured structured heat map. And maximize the data ink radio. So really use ink to show data and don't introduce decoration that might lead to chart junk. Rely on position. We saw that this is the highest element in the visual hierarchy. And we gave examples of how the matrix layout is a better way of doing this for sets, as opposed to the Euler diagram that relies on the overlap between areas. We also look at the parallel coordinates, again as an alternative of the heat map, which uses position, and the scatter plot, a two-dimensional position visualization. And the other thing you can do is to combine 2D charts for abstract multivariate data. So we show combinations of network and the adjacency matrix to deal with occlusion, parallel coordinates in the heat map or the scatter plot, and also linked views, so ways to explore data from different perspectives. So Tufte closes his book with this quote: "Was what is to be sought in designs for the display of information is a clear portrayal of complexity, not the complication of the simple." This is a nice mantra to use for our own visualizations, I think. To find out more, you can read the articles in Age of Visual Methods. They give much more detail and examples than the ones that we had the chance to present today. I also mentioned Tufte's book, which is really a very good introduction to data visualization. It's very accessible and introduces many of the concepts that I mentioned today. And there's also a book by Tamara Manger called Visualization, Analysis, and Design. This really goes through the process of designing a visualization. And if you're really interested in making something new or becoming more involved with the data visualization field, this is really a must read. I also like to point you out to a review that was published very recently called Visualization of Biomedical Data. So they give a lot of examples of good visualizations and also what is the state of the art in particular areas of biology. What I really liked from this review is this figure 2, which effectively summarizes many of the points that I tried to make before. So what is the best ways of visualizing data, depending on the type, quantitative and categorical, not using color hue for quantitative data, and avoiding rainbow color maps and the various alternatives that you can use. So you can use this as a guide for your next visualization. So I think this is it for me. I think we have quite a bit of time for questions, and I also have a quiz at the end if we have time for that.
We have time for the quiz.
Okay.
So I think while people are thinking up questions to put in the chat box, we can put that up.
So we'll do the quiz.
Let's do the quiz. Okay,
so we're doing the quiz. This is what Melissa said. So we'll go through this. Okay, so the quiz is that what may well be the best statistical graphic ever drawn? So we invite your answers to the chat box, I suppose, together with your questions, right? Okay, so we'll do the answer of the quiz. And first I'll tell you that it has to do with Napoleon, so I don't know if this gives you any hints. So this is the greatest visualization ever, according to Edward Tufte. And this depicts the campaign, Napoleon's campaign in Russia. It's a very complex visualization, but depicted in a very intuitive way. And it has 4 dimensions, at least some people say 6. You can read about that. It was drawn by Joseph Minard, I have to say, which was, again, one of the pioneers in data visualization. You can read about it in Tufte's book, and also if you Google it, you'll find a lot of information online about that. But the other thing I would like to point out is that this has been very influential in the data visualization field itself, and it has been used as an inspiration to depict pathways. So this is an example of that, and you can find more in this URL, minardo.org. And it's an interactive visualization, actually. It's an animation, so I would like to invite you to look into that and find out more about this. So now, then we have to go through, try to go through the questions. Can you help me with the questions, Melissa?
Yeah, sure. So there are a couple of questions. One of the first ones was, are there any times when you think a hairball type network is useful visualization?
Not on the top of my head. I mean, it's quite a common way of showing things. It has the advantage that you can follow a particular path, but often the way that it was drawn is really difficult. And so basically, sometimes it biases you to select a particular point. It really has a lot of disadvantages. I think what the Cerebral tool did quite well is that they show the network and they give you alternative ways of exploring it. And what we actually do in UniProt, for example, for interactions between proteins is that we actually use the matrix, because the idea is that you really want to identify these hubs and clusters that I talked about before. And then if from that, you can start by exploring the network. So from that, if you want to explore a path, then you can go and do that. But I think starting with a hairball visualization is actually quite challenging as far as I understand.
So another question is, do you need special software to plot most of the graphs?
So for some of them, you do. So for example, the upset plot, they have the tool or9. This is the thing about the sets. The gap map, you can use Morpheus. I don't really know whether there is a particular type of software like, Office for visualization. Although there is something called Tableau, which is described as the Excel for visualizations. This requires a license, but effectively it provides different views of data sets. So a lot of people use that in the data visualization field.
I guess the key is to keep it simple, and a lot of the graphs you were showing you can still do in Excel, and a lot of people have GraphPad as well. Those kind of basic bar charts are still in there.
Yeah, I think one of the things to remember is probably not to do pie charts, especially if you go to data visualization conference. Yeah, I think many of these graphs you can do in Excel, and probably the thing that Excel doesn't do very well is that it introduces a lot of decoration. So if you just keep it basic and simple, I think we'll probably follow many of the rules, and also check the colors that you use.
So there is a question saying, do you think that interactive scientific figures like the velocity-like figures will spread along in publishers?
Whether interactive figures will spread along?
Will they become more popular?
Possibly. I think some publishers use them. They are more difficult to produce from the point of view of the person who's making the visualization. And I think also there's a question, again, as I said, you need to plot what answers your question. So an interactive view is effectively, I think, in many cases, a time series, right? Or you might be using techniques like zooming and panning to focus. So if this is useful for the story you're trying to tell, then it's a good way of doing it. But you can also use different frames. So you can use different frames of your visualization in a static way. So it really depends on what the requirements are. I didn't talk a lot about static visual. I didn't talk about interactive visualization here, which are actually the ones that we make at EBI on the web, because it was more focused on figures. But it might, depending on what you want to visualize.
So there are a couple of questions that are a little bit more specific. One is about what's the best way to display flow cytometry data? And the second is, what's the best way to display correlations between disease and gene variants? And another between gene variants and vitamin D deficiencies.
Well, I think these are quite specific, so it would be difficult to answer them from the top of my head. I suppose one important thing is to think about if there are particular conventions in your field, and how well these conventions meet the needs of your audience, right? So as I said before, for example, heat maps are used a lot for RNA-seq data, right? But it's not actually a particularly good convention in terms of the colors that people use. So it traditionally used to be green and red, which is not good. And also it relies a lot on color hue for data, whereas, as I said, you can look for alternatives such as position. So for quantitative data, generally speaking, you should look for things that have to do with position. For correlations, you could use a correlation plot, but it depends a lot again on what your visualization is actually going to convey, not so much the data that you have, but the message that you want to depict. And also if there are conventions in your area that you feel are working quite well and other people understand and they can make judgments based on that, it's probably better not to invent a new way of showing things. If you do want to do that though, if you actually have a very challenging problem, let me just say that as well. if you have a very challenging problem and you have very good evidence that existing visualizations don't work very well, and then what you really need to do is to read Tamara's book, because it goes through a process of depicting visualizations. I'll talk a bit. My actual job is a user experience designer, and you're really unlikely to come up with a good visualization the first time around. What you need to do is to try different ways and then see what works best for your audience and kind of iterate on this if you are trying to come up with something new. But you need to consider whether this is necessary or not based on the state of the art in your area.
So Nikki, can you just remind us what the title of those couple of books were?
Yeah, so I'll go back to that. So this is, as I said, this is an introductory book. It kind of talks a lot about things like data inc and the basic ways of visualizing data, really, and it has a lot of examples from a lot of different fields, including statistics. And this one is more based on visualization of data, Tamara's book. And as I said, this is the place to go if you are thinking that you can come up with a better visualization yourself compared to what exists.
And just to point out, the recent paper in the annual reviews, that is an open access paper. So if you're not ready to go and buy a textbook just yet, this could be a good place to start.
That's just true, actually. It's better to start from that, particularly if you work in a particular area of biology, because they review the state of the art in each area and the advantages and disadvantages of each of them.
So we're going to leave it there for today. All of the pictures that we've used in this webinar were used with permission of the authors or under a Creative Commons license. So if you do want to reuse them, please make sure that you do that as well. If you're interested in catching some of our upcoming webinars, you can find those on our website, which is shown on this slide. So that's all from us today. Thanks very much for joining us and enjoy the rest of your day.